Hi everyone, I am Bhavna Kaur. Today I am going to present on the topic nanotubes in intercellular communication. This presentation is submitted to Professor Park in the partial fulfillment of the requirements for ELG 7132 Topics in Electronics 1. So, looking at the contents, in this I will discuss about what is cellular communication, how cells communicate with each other and the processes involved in this communication. Further, what are the basic elements involved in the interaction between the cells. I will look into what are tunneling nanotubes and how these nanotubes are formed and what causes their formation. Talking about tunneling nanotubes, I will discuss some important functions of TNTs in transport of materials like mitochondria, plasma membrane, and various other cellular components. Further, I will discuss how bacteria in nature interact with each other. I will also discuss some results of experiments performed in the laboratories to understand the concept of communication. Moving on, I will talk how carbon nanotubes can cure cancer, how they carry medicine to the targeted cells, and the basic features of carbon nanotubes. Then I will move to conclude the whole paper, and at last I will display the references I have followed to make this paper. So now let's talk about cellular internet. It refers to the internet formed due to communication between the cells which are closer or farther from each other. Cellular communication is essential for the growth of multicellular organisms. Cells interact with each other by passing messages and also pass cell components and signals to each other. Basically, there are two types of communication connections existing between the cells. The first one is small connections and the second is large connections. Small connections exist between the neighboring cells and the larger connections exist between the distant cells. Cells close to each other, or you can say the neighboring cells, these interact through gap junctions. And the cells which are far from each other, they communicate through tunneling nanotubes. Tunneling nanotubes are long tube-like structures. These structures were first defined in 2004 and these are made up of F-actin and it doesn't contain any microtubules. So now the first question is, what are these tunneling nanotubes? These are nanotube-like structures that ranges in diameter from 50 to 200 nanometer. These are found freely extended above substernum and are not attached to it. The existence of these nanotubes-like structures is dynamic in nature. These are highly sensitive to stress and their lifespan ranges from several minutes to 5 hours. These are filled with cytoskeleton filaments. These contain F-acetin and no microtubules. Several studies showed the connection between tunneling nanotubes, gap junctions, and stress. So, if we talk about the types of tunneling nanotubes, then there are two types of tunneling nanotubes. The first one is lesser than 0.7 micrometer. These allows movement of pathogens and plasma membrane, and the movement is unidirectional in nature. And the second one is greater than 0.7 micrometer. These are wider and allows transfer of mitochondria, endosomes, and the movement is bidirectional in nature. TNTs act as nanohighways for transfer of cargo and organelles between non-adjacent cells. Now, let's see how the formation of tunneling nanotubes take place. First, phyllopidium is extended above the surface of the cell, which makes connection with the other cells. And later on, it gets converted to TNTs, and it is usually seen in neurons. Tunneling nanotube-like structures were first described in neuronal cells. In their cells, de novo actin-driven formation of TNTs was observed. Further examination of PC12 cells and TNT formation suggested that while the majority of tubes formed by directed phyllopedia-like protrusions, a small subset were also able to form after cells previously in contact detached from one another. In the mouse neuronal CAD cell line, we were also able to observe both types of TNTs formation. However, the significance and the difference between these two modes of formation and whether they lead to various structures remain unclear. Similar to other cell types, we observed a high degree of heterogeneity in the diameters of TNT-like structures. Now, let's take a look on the major functions of tunneling nanotubes. These nanotubes help in the transfer of cellular cargoes, mitochondria, and calcium ions. Besides, passage of molecular signals, electrical signals, and mechanical signals also takes place. It also helps in the transfer of cellular components like plasma membrane and cytoplasmic molecules. Using Bacillus subtilis as a model organism, we visualized transfer of cytoplasmic fluorescent molecules between adjacent cells. Additionally, by co-culturing strains harboring different antibiotic resistance genes, we demonstrated that molecular exchange enables cells to transiently acquire non-hereditary resistance. Furthermore, non-conjugative plasmids could be transferred from one cell to another, thereby conferring hereditary features to recipient cells. Electron microscopy revealed the existence of variously sized tubular extensions bridging neighboring cells, serving as a route for exchange of intracellular molecules. For reference, you can look at the diagram. In this, molecular signals, electrical signals, and mechanical signals are exchanged between the cells. So let's see now how these TNTs can be fruitful in fighting against various diseases. Since TNTs allows transfer of components, we can use these components as a medium to transfer medicines. And these medicines can fight against the diseases. Further, preferential uptake of mitochondria through TNTs leads to increased survival and better resistance to stress. Polymeric nanoparticles can be moved through these links, which can be loaded with specific drugs. This type of example is explained in the further slides for the treatment of cancer. So now, if we talk about bacterial communication, it was observed experimentally that bacteria not only communicate with their same species, but they also interact and exchange messages with the different species. Molecules are packed in extracellular membrane vesicles which travel and fuse with the distant cells. Through these, DNAs are transferred through a tube-like structure called pilus from donor to recipient. So now let's discuss some results from various experiments. We anticipated that when two strains, each harboring a different antibiotic resistance gene, are grown together, the exchange of cytoplasmic molecules through the tubes could yield a population of cells temporarily resisting to both antibiotics in a non-hereditary fashion. That means cytoplasmic molecular exchange takes place and plasmids are also traded between the adjacent cells. If we look at the treatment of the cancer, there are three ways. One is surgery, which is very expensive and not convenient. If we go for radiation therapy, it can damage the already normal functioning of the cells. And if we go with chemotherapy, normal cells are subjected to random attacks. So due to all these limitations, we are introduced to a new concept, that is the use of carbon nanotubes. These are hollow cylindrical structures made from carbon and are of two types. One is single-wall carbon nanotubes and the other is multi-wall carbon nanotubes. These are good conductors of electricity and heat and are strong at high temperatures and acidic environments. These are more stronger than steel. These are the basic properties of carbon nanotubes. So now let's see how the medicines are transferred through carbon nanotubes. Magnetic nanoparticles containing anti-drugs, anti-cancer drugs are interwrapped into multi-wall carbon nanotubes, and with the help of external magnet, these are dragged to the locations where cancerous cells are residing, and these nanotubes destroy those tumor cells. These carriers can penetrate barriers that are difficult in conventional drug delivery. That's why carbon nanotubes are preferred for curing cancer. So now let's move to the conclusion. Recent studies and experiments prove that intercellular communication involves the transfer of cellular components from one cell to another, and non-hereditary resistance to antibiotics is also acquired in many cells. With we have seen this proof with the experiment performed in the laboratories. Carbon nanotubes are designed to carry drugs which kill the damaged cells and further helps in preventing the spread of disease. And most importantly, the factors on which the exchange of components through TNT depends are still not exactly known. So now let's move to the references. So below are the references which I have gone through in making this project. If anyone wants, they can look into these references. So at last, I welcome the questions from you, and I would like to thank you for watching this video.